Is the virus changing at all, the Ebola virus, so that's something we might find today that some antibiotic or something that is very successful with it today, that a month from now, is it, does it change that fast? So we have another problem.
Right. And this is exactly what's happening. The virus is mutating away in thousands of people in Western Africa. Now, my lab is making the images to understand how the virus works. So if you can show the blue and white GP protein movie. This is the structure of the surface protein of Ebola virus. You see that white piece? That's like thread wrapped around the blue spool. That piece of protein machinery of the virus will unspool and drive itself into a human cell. That's how the virus infects. Because we have these images, we know how it works, and we know what antibodies to target to different places. So what we're doing now is using the images and the structures that we have as a roadmap to make exactly the kind of antibodies we want to make. So we know that that blue and white molecule that you had is like a wolf in sheep's clothing. It's wrapped in a cloak of human sugar, human carbohydrate. On its way into the cell, it hijacks human enzymes to shed the cloak and expose the site it needs to drive itself into the cell. Working with a company called Zebra in San Diego, and also working with a professor, James Crow at Vanderbilt, we've been able to target antibodies against those sites. And the reason we know what those sites are is this structural work that you're looking at right now. So the blue and white is the surface of Ebola virus. The blue and orange movie is the shell that releases new viruses. And so we've been able to see, you can see those molecules transforming. What this virus does is something that we have not seen before. What you learn on your mother's knee as a molecular biologist is that a protein sequence confers its fold, which drives its biological function in this one-dimensional highway of destiny. What we've seen for Ebola virus is that it unfolds and refolds to be different structures at different times for different functions. It's a moving target. But because we have these structures, we understand what to hit. Now, the reason we have these structures is because
let me just ask one quick question. Is that, because I only have a few seconds left, is that, can it change so that if you do find the target of the site, so you do figure that out, is it possible that we're going to get another strain and all of a sudden we get another problem?
Well, we've anticipated this. So we're trying to come up with what I like to call Plan B map, which is the next set of antibodies we need in case the virus changes and how it responds. And that work is all supported by the National Institutes of Health. And so one thing your viewers might want to know is how this works. I mean, the free market will never support an Ebola virus therapy because usually it's too rare. The people that get it are too poor. But the reason that we have these images and we know how to fight it is because the NIH has been supporting my work in anticipation that there might be an outbreak. Now, this is a tremendous boost to the economy. If the NIH budget for a year is $30 billion, it generally makes $68 billion of economic activity. So 150 or 200% boost to the economy from doing the research that generates cures. And some projects, like the Human Genome Project, that was a $4 billion investment that resulted in $800 billion of economic activity. That's a 20,000% increase in return. I mean, what other investment can you find where you get that kind of return?
I think right now, Professor, the Americans would like to invest a lot of money to cure this because it feels very real to so many.